Hello biologists. This is a video going through the concept map or drawing that we produced in class today. Part 1, this part is about how the virus can be digested by the antigen presenting cells, and we'll finish at the point where they travel to the lymph node to try and present the antigen to the naive B and T lymphocytes. And then the next part will be about that antigen presentation process, activation of those naive B and T lymphocytes, and then their response in the third line of defense, or the specific or adaptive immune system. So let's jump into the first line of defense. So this is, when we talk about the first line, we're talking about barriers to pathogens, to prevent pathogens entering the body. So the pathogen that we've used in this example is a virus. And you can see here the most obvious barrier to a pathogen is the skin. But there are other barriers that you can discuss. So you can talk about other physical barriers such as mucus and cilia, or hairs even, fluids such as in the eye or urine, or also physical barriers because they physically remove the pathogen. And then you have the chemical barriers, the enzymes, the pH, that also limit the ability of a pathogen to actually invade your body cells. Once this has been breached, however, we initiate the second line of defense, so the innate immune system. And there are a number of things involved in that you need to be able to describe. So, here we have neutrophils, which are the first phagocytes that a virus is likely to encounter. They're the most prolific, so the most common phagocyte, and they have this distinctive nucleus which you do need to be able to recognize. Now, neutrophils are most likely to be able to engulf lots of pathogens. They don't last very long, and the death of neutrophils and associated pathogens are what cause pus to be a greeny colour. Two other phagocytes on the scene. Here we have a macrophage and a dendritic cell. Now, the macrophage is larger and not particularly distinctive in its shape, but the dendritic cell does have those long finger-like projections that engulf the pathogen, which here we have a pathogen being engulfed, should be coloured orange to indicate it's the same virus. What I would also like you to be able to stitch into your story is mast cells and their role in initiating inflammation. So histamine is contained within the mast cell. So a mast cell is a granulocyte, and the one that is mentioned in your study design. So when you sketch it, put lots of vesicles inside and note that they contain histamine, which is a tiny molecule, a signaling molecule, modified amino acid that will be released during a process called degranulation. And that degranulation will occur because of the presence of a pathogen, so that molecular pattern will be observed, or because of damage, so necrotic damage and lysis to cells due to an injury. The release of histamine here will have a number of different effects, and they result in what we call inflammation. Now, the effects of histamine, remember that we said can be summarized as A, B, C, D. So the A refers to the attraction of phagocytes. So that will cause phagocytes to come to the site of the injury or the infection. How did that happen? Oh, there we go, visiting puppy. Go away, dog. The B stands for the blood vessels becoming leaky. The C stands for the constriction of respiratory passages or airways. And the D stands for dilation or vasodilation, so the increase in size of your blood vessels to increase the blood flow to the area. And the observable symptoms of inflammation are things like redness, swelling, heat and pain, as indicated down here. You might get hot, swollen areas where there's a splinter, for example, or some kind of damage. So the histamine release does attract these phagocytes, so we're trying to recruit this second line of defense to the site. In addition, the complement system will be activated. So remembering that complement proteins are circulating, they're distributed throughout your tissues, and they will activate on the presence of a pathogen, in this case a virus. And they will do multiple things to eliminate the virus itself as well. One is opsonize, so flag the virus so it's more easily detected by phagocytes. Two is puncture the membrane in cellular pathogens such as bacteria. And three is actually act as a chemoattractant to again attract phagocytes. So we're multiple ways to get more phagocytes to the site. And if you think there is just one virus, think again. There are billions and billions of viruses that are going to get in, and it will replicate very, very quickly in the tissues. So this is happening on a much larger scale than we're representing here in this diagram. So once the phagocytes have been encouraged to attend by multiple different ways, different strategies, such as histamine complement system, they will start to engulf the pathogen. And once they've engulfed it and enclosed it in a phagosome, broken down the pathogen, the APC phagocytes, so the antigen-presenting phagocytes, being dendritic cells and macrophages, but actually also B lymphocytes, which we'll talk about later, they will break down that pathogen and then present the molecules from that pathogen on the MHC2 markers, which are unique to these antigen-presenting cells. Also present on their MHC1 markers, which are the markers showing self. So all of your nucleated cells have MHC1 markers, and you will actually also see traces of antigen on the phagocytes' MHC1 markers. Once that has happened, they will circulate through, in this situation I've put that they're travelling through the lymph, through the lymphatic system, through the lymph vessels, and they're going to arrive at the lymph node. Now the lymph nodes are not the only place that antigen presenting cells will encounter lymphocytes that they need to present to, but it is the most highly concentrated area for APCs to encounter the correct lymphocyte. Okay, so you can find lymphocytes elsewhere, so such as in the lymph itself. We might finish this part of the video there. So we've just covered the first line of defense and some aspects of the second line of defense, and what we have is the antigen presenting cells actually holding on to that specific antigen on their MHC2 marker and travelling to the lymph node, where they hope to encounter a lymphocyte with exactly the corresponding receptor. Now move on to part 2 if you are ready.